Hello. In today's lecture, we'll be discussing early clinical trials. These are very important studies. They set the stage for the more definitive registration pathway trials that may lead to the approval of a new treatment for patients to improve health. In order to understand the context in which early clinical trials can be found, we need to take a step back and think about the development process of drugs in a larger sense. I'm depicting this process as a funnel. On the left-hand side, we begin the process of developing a drug, and on the right-hand side of this funnel, the drug has actually been approved by regulatory authorities. There are many steps that occur between the left and right-hand side of this funnel. The funnel is divided into several phases. First, on the left-hand side, is the drug discovery phase. This is followed by the preclinical phase, then a whole series of clinical trials, and there are phases of these clinical trials. Today we'll be focusing on early clinical trials. We should also mention that once a drug is approved, it is our obligation to evaluate how it is operating when it is used in a broader general clinical context than we had the opportunity to study when clinical trials were performed. This is referred to as the post-approval phase. I also show on this slide the daunting task that individuals who are developing a drug must face. They need to screen about 10,000 compounds and pass it through the various steps of the funnel, and ultimately one will be approved for clinical use by regulatory authorities around the world. This development process may take as much as 10 or 15 years, and it is estimated that it can take as much as $1 billion of research and development funds. We're going to follow a white powder as our prototypical new molecular entity or drug that we wish to evaluate. That's shown on the left-hand side of the screen right underneath the drug discovery phase. And let's imagine for a moment that this white powder was one of those 10,000 compounds that had been screened. Before we can consider testing this new drug in human beings, we have to do some preclinical work, and we're representing this by our laboratory animal. The clinical translational science research spectrum has been divided into translational blocks, which traditionally have been considered to prevent an interesting observation at the bench from making it into clinical practice, a bench-to-bedside block. These have been divided into domains. And this is referred to as the T0 domain of clinical translational research, because we haven't even begun to give our interesting new compound to human beings. So we're at the T0 phase. Now, once we do our first in-human study, or FIH study, or our proof of concept study, our POC study, we've entered the T1 domain of research in our clinical translational research nomenclature. Notice that that first in-human study is that moment in time when we have crossed the boundary from the preclinical phase to human testing. We are very interested in large multi-center studies which form the T2 research domain, and it is at this point that we are actually making the transition to patients who have the disease of interest, and we're trying to study whether or not our new treatment would be safe and effective to be brought into clinical use. It's on the basis of one or more of these large phase 3 trials, which I'll discuss in a moment, that we have a situation where there is approval by regulatory authorities. Once the drug is approved, we have to understand how it can be translated into practice and how it can be used to improve global health. Researchers in this area are operating in the T3 and T4 research domain. Now I'm going to layer onto this diagram another nomenclature scheme which actually existed before the clinical translational research scheme was introduced. This scheme identifies the phases of clinical trials. And you can see that there are 4 phases. Phase 1 corresponds to the first in-human study. Phase 2 studies are proof of concept studies, and sometimes they're at the early portion of T2 research. Phase 3 studies are those that are the definitive registration pathway trials that regulatory authorities look at to determine whether or not a drug should be approved for use. And phase 4 studies are those that occur after a drug has been brought into clinical use, and these are the post-approval studies. When we think about the preclinical safety testing, our major objective is to evaluate potential toxicity of our new molecular entity, our new drug, the white powder in this case being exposed to a laboratory animal. There are 3 target organ systems that we absolutely must evaluate when our laboratory animal receives the new molecular entity. These include the cardiovascular system, the neuromuscular system, and the respiratory system. Depending upon what that drug does, there may be other target organ systems that need to be evaluated, but these 3 that are shown on this slide are absolute musts when we're doing our preclinical safety testing. Our goal here is to understand dose dependence. A term that is used is exposure dependence. Does any toxicity that appears in any of these target organ systems begin to appear as we give more and more of the drug to our laboratory animal? So-called exposure dependence, that is, exposing the drug to increasing concentrations in our laboratory animal. One of the other interesting questions that we must evaluate is if we do observe toxicity, is there potential for reversibility? Just because we see toxicity doesn't mean that the drug cannot be evaluated any further. We need to know whether or not there is a potential that the toxicity could be reversed. All along, we're assessing the physiologic response of the target system to the new molecular entity or drug of interest. There are other toxicity studies that we need to perform before we can move on to our early clinical studies. These include genotoxicity. Is there some genetic abnormality that occurs as a result of exposure of this drug to our laboratory animal? Does it produce cancers? Is there carcinogenicity? What about local intolerance? If this is a drug that's going to be given by the cutaneous route, is there cutaneous intolerance? That's an important thing to know. Phototoxicity. Do individuals who might take this drug in the future have to worry about whether or not they're exposed to sunlight where there's ultraviolet radiation? We also need to focus on whether or not there's any reproductive toxicity that's caused by this drug. The toxicity studies that I'm going to discuss can be divided into 2 broad categories. One is an acute toxicity study, where we give escalating doses to our laboratory animal until we see a serious event, perhaps the animal died. This determines the effects of an overdose. This is extremely important because, unfortunately, sometimes patients who might take this drug in the future might make a mistake and take too much of it. We have to understand what the effect of an overdose of our new drug might be in our test subject. Now, in reality, most patients will be taking drugs in a repeated fashion, over days to weeks to months, or even chronically. So we need to also have a toxicity study that's done in a repeated dose format. That's diagrammed by the multiple images of our vial of white powder, and we compare the toxicity findings in the repeated dose experiment with those that we see during the acute phase toxicity experiment. We wish to determine in our repeated dosing experiment the maximum duration of a clinical trial. There is a relationship between how long we have exposed a laboratory animal in a repeated dose fashion to our drug of interest and how long we are allowed to actually test human beings with this new drug. So it's very important that that repeated dose experiment be conducted before we do our very first human experiment. Now that we've determined exactly how we're going to proceed after our laboratory testing, we have a very important question to face. What is the maximum human recommended starting dose? There must be some relationship between the dose that we gave to our laboratory animals and that dose that we're going to start off in humans. This is referred to as the MRSD. And I'm going to show you the steps that are involved in determining the maximum human recommended starting dose. So imagine for a moment that we've given a particular dose of our new molecular entity, our drug of interest, to our laboratory animal, and as shown by the check mark, there are no toxic events that have occurred, no adverse drug reactions have occurred, and we're comfortable that the animal has tolerated that dose. Now, as illustrated by the larger size of the picture, we've given a larger dose to our laboratory animal, and now we do see a toxic effect. We move backwards in dosing, down one level, to the point where we had no observed adverse events. The dose is referred to as the NOEL dose, at which point there is no observed adverse events. That NOEL dose is going to be very important to us. Now we need to understand how we're going to make the translation from the dose, the NOEL dose in animals, to the actual dose that we're going to start off in when we go into clinical investigation. The equation, which illustrates the human equivalent dose in milligrams per kilogram, is shown on the bottom of this slide. The animal NOEL dose in milligrams per kilogram is noted, and we adjust for the body surface area of the animal compared to a human by noting the relationship in the body weight of an animal to a human and adjusting for the body surface area relationship. And this equation gives us the human equivalent dose. Now, when we start testing in humans, we need to be particularly cautious. So we don't usually start with the calculated human equivalent dose right away. We introduce a safety factor. Typically, we apply a 10-fold safety margin, and we divide the human equivalent dose by 10. So we're moving up very cautiously as we enter our first-in-human experiments. The clinical aspects of our first-in-human study are summarized on this slide. Note that it's usually a single dose to healthy volunteers. We're not talking about patients with the disease of interest. These are healthy volunteers, the very first humans to actually receive dosing with our drug of interest. Because we don't really know for sure how they're going to respond, this first-in-human study is typically performed in a controlled environment, in an inpatient environment, where very thorough and rigorous monitoring can be conducted. The three major things that we focus on in a first-in-human study are safety. Any of the toxicity studies that we examined in laboratory animals, are there any safety concerns that are starting to emerge in human subjects as we expose them to the drug? And then we're going to study pharmacokinetics and pharmacodynamics. I'm going to explain what those terms mean in just a moment. The specific first-in-human issues can be divided into two broad categories. We have to consider the route and rate of administration of our drug of interest. Is the drug going to be given intravenously? Will it be given as a bolus? Will it be given as an oral suspension? Or are we going to convert it to tablets or capsules, which we will have our human subjects swallow? We have to consider the endpoints. Surely we're interested in cataloging any adverse events that might occur in our human subjects as they receive this test drug. We also want to understand the physiologic effects of our test drug on human subjects. Now we're going to talk a moment about pharmacokinetics and pharmacodynamics. To make this crystal clear, consider these two pictures. We have the human subject on one side and the pills on the other side. When we talk about pharmacokinetics, we're considering what the human subject does to the pill once it is ingested. How well do they absorb it, metabolize it, and excrete it? When we talk about pharmacodynamics, we're talking about what the pill or drug does to the human subject. So think of it this way. Pharmacokinetics are what a human's body does to the drug, and pharmacodynamics is what the drug does to a human body, its physiologic effects. We are going to make some very early and limited observations about efficacy. Remember that we're generally dealing with a relatively small sample size, and as you've heard from other lectures in this course, in order to be absolutely confident about efficacy, we need a much larger sample size than would be the case in first-in-human studies, but we are going to take the opportunity to make some early efficacy observations. let's understand a little bit more about pharmacokinetics in first-in-human studies. So here we have our pills being ingested by our human subject, and periodically we take blood specimens. Those blood specimens could be obtained at various time intervals after our human subject has swallowed the pills. And we'll make certain measurements that are illustrated on this graph. Typically, we look for the concentration, that is the maximum one we observe after a human subject has swallowed the pill. This is referred to as Cmax, and it is seen along the y-axis. Notice that this curve is plotting the concentration over time, so we can easily identify the time it took to get to that maximum concentration, and that's Tmax. We are also very interested in the area under the curve, or the AUC, and we're also interested in how long it takes a patient to excrete or clear the drug from their body. This is referred to as the T1/2, or half time of elimination. That's defined as the time it takes for the concentration to fall by 50%. So on this slide, you can see the level of Cmax, and then about 50% of that Cmax, you can see a horizontal dashed line, and the time it takes to fall to that level would be the T1/2. We could actually do this kind of experiment in multiple healthy volunteers, and we could combine the information to obtain initial estimates of what is referred to as the population pharmacokinetic profile of our new drug. We're going to relate the pharmacokinetic parameters that are listed on this slide to toxicity and to any physiologic response that we observe. Now, once we've finished our first-in-human studies, we turn to our proof of concept studies. There's a very important difference as to who our research subjects are at this point. Once we have gained that degree of comfort from our first-in-human studies, our study subjects are now patients with the disease of interest. What are the questions we ask when we're doing a proof of concept study? We continue with our exploration of dose response. We continue with our observations about safety, pathophysiologic impact of the drug on our human subject with disease is very important to us. For example, if this new drug is an anticoagulant, we would want to study the impact of it on our patient's coagulation system. We're also going to make some limited observations about efficacy. Remember, our sample size is still relatively small here, but we are going to take the opportunity to make observations about efficacy. Now, the early clinical trials that I've just discussed require some very particular statistical analyses. And in the next lecture, you'll be learning more about those statistical issues related to early clinical trials.